Example of narrow spectrum antibiotic is penicillin. So we are going to study penicillin little in detail. Penicillin is an antibiotic which is produced by a fungus, Penicillium. Penicillium is a fungus which when grows it produces penicillin antibiotic. Okay, and this antibiotic is used to kill harmful bacteria like staphylococci, clostridium, streptococci, etc. That means only this particular group of bacteria. So penicillin comes under narrow spectrum antibiotic. Now medicines, see, penicillin cannot be directly used as a medicine. There has to be some base. When medicines are produced, penicillin will be the main principal ingredient, and there will be some other supporting chemicals with it. So when medicines which have penicillin, they are used for certain infections like ear infection in the ear, infection in the throat, nose, infection of the skin, and in case of pneumonia or scarlet fever. These are particular specific diseases in which medicine containing penicillin is useful. This photograph is of Dr. Alexander Fleming. He was the scientist who first discovered the antibiotic penicillin, and he did further work and research, and he found out that penicillin is a medicine antibiotic and it is produced by a fungus named as Penicillium. So we are going to see his experiment and how he discovered it. So here's in short a story about Dr. Alexander's finding about penicillin. So actually Dr. Alexander Fleming was a professor of microbiology, and he was doing a lot of study on bacteria, fungi, different microorganisms. So one day in St. Mary's Hospital, he was cultivating culture variety. He was studying some bacteria and fungi. And when you want to see, whenever you want to study some microorganism, any type of microorganism, you have to allow it to grow. And to allow it to grow, you have to use petri dish. So in the year 1928, on 3rd September, when Dr. Alexander Fleming was studying Staphylococci is a bacteria. When he was studying the culture of Staphylococci, he saw that the petri dish in which Staphylococci were grown, around in that petri dish, there was a fungus grown. Okay, in the petri dish, fungus was growing, and around the fungus, in round circular patches, there was some area which was clear, and then away from that particular area, the bacterial colonies were seen. What does this mean is, suppose there's a dish, petri dish. Okay, it's a dish of glass in which the organism grows. So there will be a culture means the food of microorganism. In this, he saw that some fungus was growing in some particular area. For example, this is fungal growth, and around this fungus he saw that up till a particular area, there was no growth around this fungus up till a particular distance, and away from that he saw that bacteria were able to grow. Staphylococci or some other bacteria, they were able to grow. He was not able to find out why this fungus was not growing near this, why this bacteria was not growing near this fungi. Okay, why a particular area around this fungus was clear. So to find out this, he did further studies and he analyzed this. Then he started he started studying that particular sample which was containing fungal growth. Then he found out after his studies that the fungus which had grown was Penicillium. The name of the fungus is Penicillium, and this Penicillium, when it was growing, in return it was secreting some substance which was destroying the bacterial colonies, which was killing the bacteria. So bacteria were not seen in that particular area in which that secretion was there. Then later on, research was done, and it was found out that it is an antibiotic, and the name was given penicillin. So Alexander Fleming is responsible for discovering penicillin, and there is huge tremendous use of penicillin which we have just saw in the above paragraph. So it's a tremendous and huge big discovery in the field of medicine.